Hi, this is Dr. Ben Morrill. Welcome to episode 22 of Reptile Genetics Weekly. We have a fun one, but also one that's going to require a little effort on your part if you're willing to do it. So back to school. We've got kids going back to school and figured it'd be a good time for all the DNA nerds to get back to school too. And so those of you that are interested in understanding a little deeper what's going on with genes and alleles and linkage and crossing over and all these kinds of things, we want to hit some of those subjects and then be able to use that foundation to be able to talk about things like banana ball pythons. So I think by this point, many people have figured out, how to expect when you breed a male maker or a female maker. But we want to be able to lay the groundwork so you can understand at the DNA level what's going on. And so for those of you that are willing to put in a little effort to learn, we'll try to make it as interesting and hopefully have some funny parts to it to help get you through the learning part. But I think this will be something that many of you will appreciate. So we'll give it a try and let us know in the comments. Let us know if you want more like this or if you want us to go back to other things we're doing. Let us know in the comments. We definitely read them and listen to them. But for updates, for samples, actually, there is another cool thing I wanted to mention too. For the DNA, the desert ghost testing, we had all those hundreds of sheds come in. So the 3 months is going to be up in 3 weeks from today. So on September 26th, that's when we need all of the sheds to be in for the free desert ghost testing. And since we're kind of getting out of the summer and into the fall, the other paid samples are starting to slow down some. And so by the end of this month, I expect that we will finally be running some of those free ones. I know many of you have asked several times, but we're finally to that point where we'll have all the sheds here, and we will also be able to start running some of those. So that's all coming up real quick, and we'll hopefully have some cool episodes going over that data as we have pictures and data to look at. That'll be really cool. So yeah, remember, any of you that have not sent your shed in yet, hopefully you've already sent in the picture and the information, but just getting the shed in, we need that by September 26th, 3 weeks from today. All right, so for updates, good news and bad news. I'll start with bad news. The big run I was going to do this week, there's several, a fair amount of it failed our quality control at the beginning. So thankfully it's very early on, so nobody has any results that are incorrect or anything like that. We have several steps where we do quality control. And at the very beginning, we had several fail our quality control. So this week, either this weekend or on Monday, hopefully at the latest, we will have all the panels that came in over about the last month or so. The full panel, the way we ran them with the full panels, those seem to work just fine. They look good so far. And that will probably be it. So if you are doing like a single gene or a 2 gene and you are expecting to have that on this run, unfortunately, it's going to get pushed back a week. But the good news is we are going to do a lot of runs between now and Tinley, October Tinley. And so for the next probably 3 or 4 weeks, we'll be doing a run every week. So those results will just get pushed back 1 week and we'll be on them. And next week, I expect to have a couple of new tests that we'll be able to have available. And the week after that, another one. And then probably another 1 or 2 or 3 almost every week until Tinley, I would bet. Maybe we'll miss a week here or there, but we've got a lot that are right at the finishing line. So we got a lot of cool stuff coming. But anyway, I got Kayla here with me today. I also got Travis. How y'all doing?
Hey, guys. Doing good. Glad to be here.
Ready to go back to school?
Yeah.
Go back to school.
So once again, Kayla will help represent the people that aren't super nerds like Travis and I, and have been thinking about this for 20 or 30 years. So we might quiz Kayla a little bit as a way to test and see if we're explaining this well.
I didn't study. Let's go.
Best way to do it.
Yeah.
That's right. So we're going to talk about how to make a reptile. So you could think about DNA as being the blueprint or the instructions on how to make a reptile. And it's a whole bunch of A's, T's, C's, and G's. So in our language, we use 26 letters, the alphabet. But for DNA, there's just 4 letters, A, T, C, and G. And so all that coding to make an ear, a foot, liver, all of those things is all coded in this same 4-letter language that's saved in the DNA. And instead of kind of like if you were going to have a website, you wouldn't have everything just on one page or you have to scroll and scroll forever and ever. You're going to break it up into multiple pages. And that's what chromosomes are. So for all of that information in the DNA to make a reptile is broken up into chromosomes. And in snakes, there's 18 of those. Did we hit everything on that slide?
Yep, you got it. And you just touched on the second one.
All right, Travis, you want to take this one?
Sure. So when we're looking at snakes, they've got 18 chromosomes worth of the blueprint information. So you need to make sure when you are making your snake that you reproduce those chromosomes and the information within them. And basically, so that's your formation of your gametes, your sperm and your eggs. So you've got to take those chromosomes, you've got to package them up and send them away. And you've got to make sure you have 1 through 18 in each of those little packages. And there are a bunch of different ways you can do that. Now, if you're not a reptile, if you're one of the simpler organisms on the planet, and I say simple only in as much as how they do things, not so much DNA, because amoebas have a huge amount of DNA compared to almost everything else out there. They just reproduce by sexual reproduction. Basically, they double themselves and then split in half and boom, go off, and now you have 2 of what you started with. You can't really do that with a snake. A snake can't just split down the middle and suddenly turn into 2 snakes. This is good for static environments. Places where there aren't a lot of major changes or where you don't have a lot of disruption to your system, because you're all clones of yourself. And so anything new comes along that you're not familiar with, your whole population will collapse and crash.
Yeah, so that's asexual reproduction. And then this next slide, we'll talk about sexual reproduction. I love these are those, what species are those, Kayla?
Oh, those are garter snakes. I don't know which flavor Thamnophis they are, but
garters in love. So we have sexual reproduction. So it does actually take 2 different individuals. And so if you're in an environment that's changing, which is the environment that, if you're a medium, even a small, medium to large animal, for a really small organism like an amoeba, maybe you can be in a spot that doesn't change a lot. But if you're an animal, and even plants, if you're big enough, you're likely going to be in an environment that's changing. There will be things that will not stay the same. And so you need to be able to evolve. You need to be able to change. And the way you do that is genetic variation. So a lot of the time we'll hear, in the news or whatever, we'll hear about a new species or, splitting, and we hear about it in herpetoculture as well. People that want to split one species into 2 subspecies to make sure that they properly protect, or it could be even splitting one species into 2 separate species. You want to protect the genetic variation that's there and make sure it's named and protected. So with sexual reproduction, you have 2 different individuals that make a gamete, like Travis was talking about earlier, a sperm or an egg. And each one of those has 18 chromosomes. So you get 18 chromosomes in the sperm and another 18 chromosomes in the egg that come together. So you have a total of 36 that are paired chromosomes. 18 came from dad, 18 came from mom. And let's see, I think we're ready for the next slide. Yeah, so here you can see the sire we have there. It's red and gold, I guess.
Yeah.
So that sire is made up of his mom and his dad, his sire and dam. And so he's half sire, half paternal grandsire, half maternal granddam. And then when he makes sperm, what's going to happen is he could pass all, like the one in red there, he passed all of his genetics from one of his parents, his red parent, in one. And the gold sperm there, he passed all of his genetics from the other parent, so it's all gold. And that's very unlikely to happen because it's a 1 in 2 chance for all 18 chromosomes. So it's 1/2 to the 18. That's a very, very big number. Very unlikely that you would do that, but it is possible. And then all the combinations in between. You could have you could have all the odd chromosomes come from mom, all the evens come from dad. You can have just any combination of that. And that's what gives animals and plants that are made from sexual reproduction, gives them a lot of the genetic variation that they have. Mm-hmm. All right, Travis.
This is easy for me. So basically, you get the same thing going on with the mom. Then the dam is sired from her maternal father or her maternal grandfather and her maternal grandmother, and they give her again 50% each. And so when she develops her eggs, again, you can have the possibility where she has an egg that carries all of the genetics from her father or all of the genetics from her mother. But you're a lot more likely to get the egg where it's a mix of, 50-50 or 70-30, 60-40. There's no exact number. We always say 50-50 in, the basic education world because that's where it falls in the bell curve.
Yeah.
Now, what does happen is you will get 50-50 from, so that sire, any offspring from that sire are going to have half from him, and then any offspring from that dam are going to have half from her. But then when you look at their sperm and egg, that's where we're talking about there could be this variation from 0 to 100% from one either maternal or paternal grandparent. So that's where you get that big mix, is because each sperm and each egg is potentially a big mix. But then when the sperm and egg meet, if you look at it just from that sire and that dam, yeah, half the genetics is coming from the sire, half is coming from the dam. But all the generations before it provided all that genetic variation so that you don't have, like here we have the number, it's 1 in 262,144 chance that any two gametes would be identical. And that's before they combine with each other. So that's talking about 2 sperm being identical or 2 eggs being identical. That's 1 in 262,144. So it's very unlikely. So Kayla, so far, how's it looking?
So far, I mean, seems simple enough for me. I did do the little doodles and tried to make these visuals that would represent what I got when we were making the slides. So I think for this one concept, I think it should be pretty easy to pick up, I think.
Good. As you were preparing, because Kayla did a beautiful job with these graphics. As you were preparing and thinking through these, what were the concepts that were kind of the ones you had to think about the most?
I think the one that I had to think about the most was, honestly, the thing that jumped out to me was the fact that theoretically there was a chance for, if we have the maternal dam, so, the purple snake in this visual here, the dam could, in a way, pass down like only her mother's DNA in one gamete, which is bizarre to think about and also extremely unlikely, but there's the chance for it. And so if you think about, you see the one purple egg in this drawing, that could mean that and when I made this visual, I actually did follow the arrows when coloring the snakes in the next slide. So, I mean, you could have on the left side, that purple, gold, and red snake that doesn't have any of the cyan DNA from the maternal dam. And I thought that was wild. And it kind of clarified just how much genetic diversity does come just from the like just from this process alone, of splitting up DNA and passing parts of your genetic information through gametes.
And that's a great observation, and I would like to use that as a small pivot point. So if you go back to the group picture of everybody,
with all the
imagine if we're dealing with the dwarf or the super dwarf retics, and you've bred it to a mainland. Now, a lot of people, they'll breed two 50% dwarf or super dwarfs together, and they'll still label it as a 50% because technically, mathematically, that's correct. But imagine if the genes that gave it that dwarfism were all located on that cyan, the 18 chromosomes that were cyan. So that purple, red, and gold snake wouldn't have any of the dwarfism genes to it, but people would still label that as a 50% dwarf.
Wow.
And so
I didn't think of that.
And a lot of people in the hobby don't think about things, that way. Yeah. So it's, the math works out. The math sometimes lies to you. Yes, 50% of that animal, is from that original pairing, but you didn't get any of the genes. So it's entirely possible that 50% dwarf will grow to huge proportions. Yes. Because it's not actually carrying any of those dwarf genes. And I think a lot of, like Garrett in his breedings and stuff has, he's played that deck in the opposite direction. He breeds the smaller animals that he knows are carrying those genes. And those are the ones that he calls, says that have the dwarf or the super dwarf phenotype. But if he gets one that's really gigantic, he's not going to label it as a super dwarf because he knows that, it's not behaving like a dwarf or a super dwarf. So even if it may have the mathematic blood to say it is, it turns out that the phenotype is very much away from that because it never actually inherited those genes.
Yes. And one other quick concept I'll throw in there, one that I talked with Garrett about a little bit. He definitely understood, but I think a lot of people don't recognize, and this has been pointed out in dogs and other species as well, but when you first do a cross, like if you do a super dwarf to a mainland, what you're going to get is a whole bunch of animals that look similar because you're essentially making it heterozygous across everything. And so you'll get an F1 that's pretty uniform. So that first group, they'll all be similar to each other. But then when you do the breeding like Travis just brought up, if you breed two 50%ers, so half mainland, half super dwarf, you breed those two together and you get what you call the F1 explosion. And so then you end up with like the picture, if you want to bring that picture back up again. Yeah. You didn't know how popular that picture was going to be, did you, Kayla?
No.
So
that's awesome.
This would be an example of when you breed two 50%ers together, you get that F2 explosion. You get all kinds of variation. You can get some that look and have the traits of just one parent. You can get some that look and have just the traits of the other parent, and then you can get everything in between. In that first breeding of a mainland to a super dwarf, you will not get that. It'll be uniform. The second breeding, where you breed two F1s together, where they're 50-50s already, you're going to get a lot of variation.
Huge variety. Using your dog example is a great one, Ben. Think about all the custom, the custom designer breeds. Now, like if you took a golden retriever and a Doberman, all those puppies, initially will look the same. But if you take two of those puppies and breed them together, like in theory, you could get a perfect Doberman on one side and a perfect golden retriever on the other side. But in between, you're going to have every kind of mix. Like it could look like a Doberman exactly, except it could have the coat color of a golden retriever. Or it could look like a golden retriever, but with the coat color of a Doberman. And every blend in between there, that's that explosion of variety that you see.
Yeah. Yeah, and normally you don't think about that sort of thing happening, but the other thing that kept coming to mind when I was making these visual examples was this is only zoomed in on the dam or the sire. Whereas in reality, both of the maternal and paternal parents are their own mix of colors. And so you're getting to deal with basically this mix over here before each generation. And that's a lot.
Yes.
This is why we get variety, and why every normal ball python is not a dinker just because it looks marginally different from its brother. Of course it's going to look marginally different from its brother. They're all clown poos.
Yeah, exactly.
That's right.
All right.
Excellent.
Oh, cool.
And that genetic variation is what got us so we could have an opposable thumb and a brain that increases in size, and all the variety we have in living organisms on the planet is because of genetic variation and then that evolutionary process. So yeah, hopefully that's helpful. Not too nerdy, I hope.
I mean, even for someone who thought previously that I had, sexual reproduction, one from one half, one from the other, I think I've got that because, I work with recessive gene hognoses and rat snakes. I thought I had a decent grasp of it, but just this alone, in learning about it and making the visuals for it, a lot more of the complexities were illuminated for me. So I hope that other people find this to be a similar experience. So
yes. And next week we'll add in crossing over, and we'll talk about bananas and sex linkage. I think people will find that pretty interesting once you have this foundation. But like I said, let us know in the comments, even in this video, but especially next week after we do the second video. Let us know if you want some more like this, where you learn a little bit more in depth what's going on in the DNA, and we'll give you more.
Yeah.
Yes, for sure.
All right. Well, that was a nice short and sweet episode. I hope everybody liked it. As always, make sure you like, comment, subscribe. It helps us with the algorithm and introduces us to more people in the hobby. Share with your friends. For any sciencey teacher people, if anybody wants the little images that I use, please let me know. I'll send you the stuff if that's helpful. Yeah, I think y'all got anything else?
No, I'm good.
Sweet.
Good.
All right. Well, thanks everybody, and we'll see you next week. Outro in 3, 2, 1.